Imagine a vast library filled with ancient texts. Each book, each manuscript, holds a wealth of knowledge, a treasure trove of information waiting to be discovered. This library is not just any library, it is the library of life itself. These texts hold the secrets to building and operating a human being. They contain the instructions for every process, every function, and every characteristic that makes us who we are. From the color of our eyes to the way our cells function, it's all written in these ancient texts. This library is our genome, and the texts are our DNA. The genome is a vast collection of genetic information, a comprehensive guide to our biological existence. It is the blueprint of life, meticulously crafted over millions of years of evolution. The genome is the complete set of genetic instructions found in every cell of our bodies. Each cell, no matter how small, contains a full copy of this genetic library. It's like having a complete set of encyclopedias in every room of a house, each one containing the entire history and instructions for the house itself. It's a blueprint, a recipe book, a history book, all rolled into one. The genome tells the story of our past, guides the present, and influences our future. It is a living document, constantly being read and interpreted by the cells in our bodies. This incredible library is written in a language of just 4 letters, A, T, C, and G. These letters stand for adenine, thymine, cytosine, and guanine, the 4 nucleotides that make up DNA. Despite the simplicity of this alphabet, the combinations and sequences they form are incredibly complex. These letters represent the chemical building blocks of DNA, the molecule that carries our genetic information. Each letter pairs with another in a specific way, A with T and C with G. This pairing is what allows DNA to replicate and pass on genetic information from one generation to the next. Like ink on a page, these letters are arranged in a specific order, forming the words and sentences of our genetic code. This code is read by the cells in our bodies, guiding their functions and ensuring that everything runs smoothly. It's a language that our cells understand perfectly, even if we are only beginning to decipher it ourselves. The human genome is incredibly long. It contains over 3 billion letters, making it one of the most complex texts ever written. Each letter is a piece of the puzzle, contributing to the overall picture of who we are. If we were to uncoil the DNA from a single cell and lay it end to end, it would stretch over 6 feet. Now imagine this length multiplied by the trillions of cells in our bodies. The total length of DNA in a single human body could stretch to the sun and back several times. To fit all this information into a tiny cell, DNA is tightly packaged and organized into structures called chromosomes. Each chromosome contains a single long DNA molecule, coiled and folded in a way that allows it to fit within the cell nucleus. This packaging is crucial for the proper functioning of our cells. Understanding the genome is like learning a new language. It requires patience, dedication, and a keen eye for detail. Scientists around the world are working tirelessly to decode this language, uncovering the secrets hidden within our DNA. It allows us to decode the secrets of life itself. By understanding the genome, we can learn how our bodies develop, how they function, and how they respond to different environments. This knowledge has the potential to revolutionize medicine, agriculture, and many other fields. It helps us understand how we inherit traits from our parents, how our bodies function, and even why we get sick. By studying the genome, we can identify the genetic basis of diseases, develop new treatments, and improve our overall health and well-being. The Book of Life is a never-ending story, one that we are only beginning to read. Think of chromosomes as the chapters in the Book of Life. Just as a book is divided into chapters that tell different parts of a story, our chromosomes contain the instructions that make us who we are. They are the blueprints of our existence, holding the secrets of our biological makeup. Humans have 23 pairs of chromosomes for a total of 46. Each pair is unique and carries specific genetic information. These pairs are like matched sets of encyclopedias, each volume containing crucial data that contributes to the overall narrative of our lives. We inherit one set of 23 from our mother and the other set from our father. This combination of genetic material from both parents creates a unique genetic blueprint for each individual. It's like receiving half of a library from each parent, merging to form a complete collection of genetic instructions. Each chromosome carries thousands of genes, like individual stories within a chapter. These genes are segments of DNA that contain the instructions for building proteins, which are the building blocks of our bodies. They determine everything from our eye color to our susceptibility to certain diseases. These chapters, our chromosomes, are essential for life. Without them, the intricate processes that sustain our bodies would not be possible. They are the foundation upon which our biological systems are built, guiding the development and function of every cell. They ensure that genetic information is accurately copied and passed down from one generation to the next. During reproduction, chromosomes are duplicated and distributed to new cells, ensuring that each new organism receives a complete set of genetic instructions. This process is vital for the continuity of life. During cell division, chromosomes are duplicated and distributed to daughter cells, ensuring each new cell receives a complete set of instructions. This meticulous process ensures that our genetic information remains intact and is faithfully transmitted to future generations. The 23rd pair of chromosomes determines our sex. This pair is unique because it carries the genetic information that dictates whether an individual will develop as male or female. It's like the final chapter in the book of life, revealing a crucial aspect of our identity. Females have 2 X chromosomes, while males have 1 X and 1 Y chromosome. The presence or absence of the Y chromosome is what determines an individual's sex. This tiny difference in genetic material has profound implications for development and identity. The Y chromosome contains genes that trigger male development. These genes initiate a cascade of biological processes that lead to the development of male characteristics. It's like a key that unlocks a specific set of instructions, guiding the formation of male traits and features. Sometimes changes or mutations can occur in our chromosomes. These mutations can lead to genetic disorders or variations that affect an individual's health and development. Understanding these changes is crucial for diagnosing and treating genetic conditions. Genetic counseling can help individuals and families understand their genetic makeup and the implications of their genetic information. It provides valuable insights into inherited conditions and helps guide decisions about health and reproduction. Advances in genetic technology, such as gene editing, offer new possibilities for treating genetic disorders. Techniques like CRISPR allow scientists to precisely modify genes, potentially correcting genetic defects and improving health outcomes. As our understanding of chromosomes and genetics continues to grow, so too does our ability to harness this knowledge for the betterment of humanity. The future holds exciting possibilities for genetic research, offering hope for new treatments and a deeper understanding of the blueprint of life. Genes are the individual words within the chapters of our chromosomes. They are specific segments of DNA that provide the instructions for building proteins. Proteins are the workhorses of our cells, carrying out a wide variety of functions. Some genes determine our physical traits like eye color, hair color, and height. Others influence our susceptibility to certain diseases. Genes can even play a role in our behavior and personality. Each gene has a specific location on a chromosome, like an address in a city. This precise organization is crucial for the proper functioning of our cells and our bodies as a whole. For a long time, scientists believed that genes were the only important parts of our DNA. They thought that the sequences which coded for proteins were the key to understanding everything about our genetic makeup. This belief was based on the idea that proteins are the workhorses of the cell, carrying out all the essential functions needed for life. However, it turns out that only a small fraction of our genome actually codes for proteins. In fact, only about 1 to 2% of our DNA is made up of these protein-coding genes. This discovery was quite surprising and led scientists to question the role of the remaining 98% of our DNA. The rest, about 98%, was initially dismissed as junk DNA. This term suggested that these sequences were useless, a byproduct of evolution with no real function. For many years, this so-called junk DNA was largely ignored in genetic research. We now know that this junk DNA is far from useless. Recent research has revealed that these non-coding regions of the genome are incredibly important. They play crucial roles in regulating various genetic processes and maintaining the overall health of an organism. Non-coding DNA plays a vital role in regulating gene activity. It helps control when and where specific genes are turned on or off, ensuring that proteins are produced at the right times and in the right amounts. This regulation is essential for proper development, cellular function, and adaptation to environmental changes. It acts like a complex control panel determining when, where, and how much of a protein is produced. This control panel is made up of various elements including enhancers, silencers, and insulators, which work together to fine-tune gene expression. Some non-coding DNA sequences act as switches, turning genes on or off in response to environmental cues. These switches, known as regulatory elements, can respond to signals from inside the cell or from the external environment, allowing the organism to adapt to changing conditions. Others act as volume knobs, fine-tuning the levels of gene expression. These elements can increase or decrease the amount of protein produced by a gene, ensuring that the right balance is maintained for optimal cellular function. This fine-tuning is crucial for processes such as growth, development, and response to stress. In addition to these regulatory functions, non-coding DNA also plays a role in the structural organization of the genome. It helps to package the DNA into a compact, organized structure called chromatin, which allows the vast amount of genetic material to fit inside the cell nucleus. This organization is essential for the proper functioning of the genome, as it influences gene accessibility and expression. Furthermore, non-coding DNA is involved in the repair and maintenance of the genome. It contains sequences that help to identify and fix errors in the DNA, ensuring the integrity of the genetic information. This is particularly important for preventing mutations that could lead to diseases such as cancer. The discovery of the importance of non-coding DNA has revolutionized our understanding of genetics. It has opened up new avenues of research and has led to the development of new technologies and therapies. By studying these non-coding regions, scientists hope to uncover new insights into the mechanisms of gene regulation and the causes of genetic diseases. As we continue to explore the mysteries of non-coding DNA, we are likely to uncover even more surprising and important functions. This research holds great promise for the future of medicine and our understanding of life itself. The once dismissed junk DNA is now recognized as a treasure trove of genetic information, waiting to be fully understood and harnessed for the benefit of humanity.